Hello and welcome to the webinar on finding significant SNPs using Avadis NGS. So as part of this webinar, we will discuss what the variant detection problem is, the need for filtering the reads before carrying out SNP detection. We'll provide insights into the SNP caller that is included with the Avadis NGS software. We'll describe what are the different attributes that are associated with the SNPs that are called out by the SNP caller. Then we'll talk about how to verify the SNPs that have been called visually. And then even when you analyze a single sample, you could end up with hundreds of thousands of SNPs. How do we go about prioritizing those SNPs in the case of a single sample? And finally, the problem gets only compounded when you have multiple samples. So how do you compare SNPs across multiple samples? So let's start with the variant detection problem. Suppose we have a reference sequence, as is shown in blue at the top. Here, for illustrative purposes, it is approximately a 28 base pair sequence. And suppose you had the whole genome sequence of an individual sample, and that is shown at the bottom. If you had both of those, then you could simply align the two sequences against each other. And from the alignment patterns, you could spot the deletions which occur with respect to the reference, the insertions that happen with respect to the reference, and the substitutions that happen with respect to the reference. However, sequencers don't output entire genomes. What they do output is a collection of reads, which are small portions of the genomic sequence. And then the aligner takes these reads and performs a comparison against the reference sequence and aligns these reads against the genome sequence, and those are what we call matches. So the SNP algorithm has to pretty much walk along the reference, looking for, at each base, looking at all of the overlapping reads in that location and make an assessment of whether those bases agree with the reference or whether they're different, and if so, in what manner are they different. For this, we need to ensure that whatever reads are there at that particular location genuinely belong at that location, and the content of those reads is trustworthy. The content of the reads is indicated by the base quality associated with each of the bases in the read. So the read quality is an average of those qualities, so read quality becomes an important factor. Similarly, even if the individual bases have high quality as assigned by the sequencer, it is possible that the aligner made a mistake and placed the read in the wrong location, and this will be indicated by the mapping quality that the aligner provides for the read. The mapping quality becomes a second dimension. So for this reason, before SNP calling is done, we need to carry out quality control, where we remove the poor quality reads and we remove the poor mapping quality reads. Avadis NGS provides a whole lot of QC views. The two most useful views for SNP calling are shown out here. One is the read quality distribution, which shows the average quality of a read across the entire set of reads in the sample. And the second plot is the mapping quality distribution, where the mapping quality is a number associated by the aligner. By consulting both of these charts, users can infer appropriate thresholds for good and bad quality reads and matches. And once we find those thresholds, we can remove the poor reads and the poor matches using a comprehensive set of filters that are present in the workflow. A portion of the workflow is shown on the left out here. We can see there are 5 different filter options. The filter by read metrics is by far the most commonly used filter, and the dialog that it shows is shown on the right out here. We can remove reads which have poor read quality. We can remove reads with ends in them. We can remove reads with lower alignment scores and mapping qualities, and those which have been indicated by the vendor to fail the QC criteria. After we remove all of those reads, we can then proceed on to SNP calling. As I mentioned earlier, when we want to do SNP calling, the SNP caller is going to essentially go to each location along the reference, consult the reads which are overlapping that particular location, and take the base quality, the mapping quality, and number of reads into consideration before making a SNP call. However, instead of carrying out the statistical analysis at every single base along the reference, we can choose to do this SNP calling only at locations which have a sufficient amount of reads overlapping them, and only at locations which have at least a minimum number of variant bases. This helps to speed up the SNP detection process. To summarize, the various factors that are considered by the SNP caller are the mapping quality of the matches, the base quality of the reads, and the coverage. We will not go into the actual statistical methods of the SNP caller. It is loosely based on MAQ and GAPK toolsets, but we'll just provide some insights. For instance, let us say that the reference base is a T and there are 8 reads overlapping this location. 4 of the reads have a T at that location and 4 of the reads have an A at that location. Then what are the different genotypes that we would expect? Logically, we would expect a heterozygous AT call at this location, but the actual call that is made depends on the quality of the bases involved. For instance, if all of the 4 A's happen to be of low quality, let us say Fred quality 3, and if all of the T's happen to be of very high quality, quality 30, then the evidence of the A's would be disregarded to some extent, and the call that you would expect to be made is a T call, homozygous T. Similarly, if the bases were of equal quality, that is, all the 4 A's are of quality 30 and all of the 4 T's are also of quality 30, then one would logically expect a heterozygous call, AT. And if the tables were reversed, if all of the 4 A's are of high quality 30 and if all of the 4 T's are of lower quality 3, then one would expect a homozygous A call. And that is indeed how the Avadis NGS SNP caller functions. What this slide emphasizes is that the quality of the individual bases is taken into consideration when making the call. Now let us say that we have the same proportion of bases, but the number of bases changes. Let us see how the SNP call would change. Suppose again we had 4 A's and 4 T's at this location. The A's happen to have Fred quality 5, which is quite low, and the T's have a high quality. Then one would expect a homozygous T call to be made, and that is what happens. However, if instead of 4 A's, we had 500 A's at this location and 500 T's, so the proportion of A's and T's is the same as the previous case, and the qualities are also exactly the same. Now we get a heterozygous AT call. That is because it is not statistically feasible for 500 sequencing errors to result in A's, so the SNP caller takes all of these into consideration and concludes that the most probable genotype is indeed an AT. So the counts also do matter. So all of these different factors, the quality, the counts, the proportions, all of them are taken into consideration by the SNP caller when making a SNP call. So let us see what the called SNPs look like. This is a single sample report. Each row out here indicates one variant allele. The first few columns indicate the location and the type of the variant. So you have the chromosome, start and end positions of the variant, the reference base, the variant base, and the type of the variant, substitution, deletion, insertion, etc., the zygosity and the SNP call, and the score that is associated with the SNP call. In other words, the confidence that the SNP detection algorithm has on that particular SNP call. And then you have supporting attributes. You have the percentage of reads which support the variant allele, total number of reads overlapping that particular location, the fraction of those reads which are different from the reference, and strand bias. So the 3 columns out here, the zygosity, the SNP call, and the score are assigned by the SNP caller. The others can be considered to be qualifying attributes. And the attributes are the total number of reads overlapping that location, the percentage of those reads which support the variant allele, the percentage of reads at that location which are different from the reference, and the strand bias. So supporting reads and variant reads are, the definitions are quite straightforward. So let's take a look at strand bias. This is a snapshot of the genome browser. We have the coverage tracks at the top, we have the reference sequence below that, and then we have the individual reads. Wherever the read sequence does not match the reference sequence, that is indicated by the bases. Here we can see that there seem to be 2 locations along the genome where the bases in the reads differ from the reference sequence. One is this location out here where the reference has a G, but you see a lot of A's in the reads also. And that fact is summarized by the mismatch histogram, which takes all of the bases overlapping this particular location and summarizes the counts of the number of A's, the number of G's, the number of T's and C's. So you can see from this mismatch histogram that you have approximately 50% G's and 50% A's at this particular location. In addition to the mismatch histogram, we also have the coverage. So we can see that there are 104 reads overlapping this particular location. The genome browser included Avadis NGS allows you to modify the genome browser views in multiple ways. One of them is to plot the coverage of the positive and negative reads. For instance, one might be interested how many positive strand reads are overlapping this location and how many negative orientation reads are overlapping this location. And those two coverages are drawn in different colors out here. One is the positive coverage and one is the negative coverage. And one can see that there's approximately uniform coverage in both the orientations. So we have something like 48 reads overlapping in one orientation. Maybe we have 54 reads overlapping in the other orientation, for a total of coverage of 104. So at this particular location, we have roughly equal number of ref and variant bases, that is indicated by the mismatch histogram. We have roughly equal number of positive and negative aligned reads. But what we don't know is how the variant reads are distributed in the positive and negative orientations. For instance, it is possible that all of the variant bases are there only in the negative strand. We don't know whether that is indeed the fact or not. So that is what the strand bias tries to estimate. A strand bias is a number between 0 and 200, where 0 indicates that the variant bases are distributed proportionally amongst the different orientation reads, and a strand bias of 200 would indicate that all of the variant bases are there only in one orientation read. To make this a little bit more clear, let's take a look at this cartoon. Suppose we have 8 reads overlapping a variant location, and we know that there are 4 variant bases amongst these 8 reads. How would we expect the 4 variant bases to be distributed? Since there are 4 variant bases and 8 reads, the probability that a variant base occurs in a read is half. So here we have 4 reads in the positive orientation. We would expect half of them to have the variant bases. And similarly, we have 4 reads in the negative orientation. We would expect half of them to have the variant bases. So we would expect the mismatch bases to be present approximately in this manner, and that would indicate there is no strand bias at all. However, if all of the mismatch bases happen to be present only in reads aligning in one direction, then one would consider there to be high strand bias at this location. Now that we've seen the different attributes that are associated with the SNPs, let us see how we would verify the SNPs visually. The first tool that we would use to verify is the genome browser itself. You can drag the SNPs that have been detected into the genome browser as a track and quickly navigate from one SNP to the next. At any SNP location, one can consult the mismatch histogram to see what is the fraction of reads that are supporting the reference and the fraction of reads that are supporting the variant allele. In addition, if you are interested in read level details, we can just place a mouse pointer at any location in the read track, and it will give us full details of the read that is under the mouse pointer and details also of the individual bases. However, if the coverage track is very deep, because you have a lot of reads overlapping that location, then it's not possible to visually inspect each one of those reads. So one can launch the variant support view by simply clicking on the right click, using the right click button on the read track. What the variant support view does is it takes a 10 base pair window on either side of the base of interest. So overall you'll have a 21 base pair window comprising of 10 base pairs to the left of the location of interest and 10 base pairs to the right of the location of interest. It takes this 21 base pair slice through the read track and clusters all the reads which have identical sequence. So we can see that here, even though there are 409 odd reads, 264 reads have exactly the same sequence as shown in the first row out here. There are 11 other reads which have more or less the same sequence except that they don't have an A, and so on. So the variant support view can take a very high coverage region and compress it to a small number of rows in a table. This is helpful for visualizing the neighborhood quality. Is there a lot of noise? Are there other SNPs present in the vicinity of the SNP, etc. So those are the two primary ways of visualizing SNPs and verifying them. Now suppose we have hundreds of thousands of SNPs in a single sample. How would we go about prioritizing these SNPs? One of the most commonly used methods is SNP effect analysis. That is, where we try to find SNPs which are having a consequence on the genes that are overlapping that particular location. So Avadis NGS includes a SNP effect analysis workflow step, which can compute the effect of the variant allele in different manners. If it is overlapping a protein coding region, it can compute the effect that the variant allele has on the transcript at that location, whether the start site is being lost, whether a stop site is being gained, whether it's a non-synonymous coding, or whether it's having an effect on the splice site, etc. We can also compute the non-protein coding effects. For instance, it could be falling in the 5' and 3' UTRs, etc. Once we run the SNP effect analysis workflow step, the effects that it computes for each variant allele are added as additional columns to the region list. So if you recall from the previous slides, each row in the SNP output, it has the location information, the reference and variant information, and it has all the supporting attributes. The SNP effect workflow step finds out what is the overlapping gene at the location, what is the overlapping transcript, and computes the effect that the variant allele has on the transcript. So here we can see the amino acid changes, the position in the protein, the position in the cDNA coordinates, etc. Now once we have a huge spreadsheet like this, one would want to filter and find out those alleles which are having just a non-synonymous coding effect, or those which are having just a frame shift effect, etc. For that, there's a comprehensive region list operations tool, which essentially takes this spreadsheet and allows you to slice and dice it in multiple manners. It includes a histogram feature, so you can take any column of this spreadsheet and plot it as a histogram. Here we have plotted the various genotypes that have been called in a particular sample. One can do overlays with a whole lot of other columns. Here we have done an overlay with the reference allele. We can also do a scatter plot to see the relationship between different numerical attribute columns. Here is a scatter plot showing the relationship between the total number of reads and the score. This was from an imported VCF file. So we can see that there seems to be a linear trend between the total reads and the score assigned to the variant call. But you also see that there are some alleles which, in spite of having a large number of reads, they get a very low score. And that is because they have high strand bias or some other negative features. So using the shape by, the color by, and the size by attribute features in this tool, one can try to figure out the reason why some variant alleles have been assigned a low score. One of the most important use cases for region list operation is to filter the SNP calls. Here, for instance, we can set the filter to say that we're only interested in those variants where the coverage is at least 100, where the consequence is non-synonymous coding, where the percentage of supporting reads is at least 40, and immediately you can see that out of 133 total number of alleles, it has now come down to 38. So in this manner, one can filter out important variant alleles from hundreds of thousands of alleles occurring in the sample. Another way of prioritizing the variant alleles is through external annotations. We include dbSNP annotations along with the software. So one may want to extract only those variant alleles which are not present in dbSNP, that is the novel alleles, or one may be interested in those which overlap with other annotations such as biobase. Biobase is an additional feature, and the genome tracks that are provided with biobase annotations are ChIP-seq sites, HGMD mutations, transfac sites, DSS, etc. So now we have a fairly good idea of how SNPs are called in a single sample, all of the different properties that are associated with the SNPs, how to filter the SNPs using region list operations and other annotations, and how to use the SNP effect prediction tool. So now if we are called SNPs in an experiment where we have multiple samples, then how do we prioritize the variant alleles? When there are multiple samples present in the read list, in addition to generating the single sample reports, Avadis NGS also generates a multi-sample variant report. The multi-sample variant report has one row for each variant allele which has been discovered in any one of the samples. So one can think of it as the union of the various single sample variant reports. But for each row out here, for each variant allele, it has 6 attribute columns derived from each sample. So the first set of 6 columns are the attributes derived from the first sample. The next 6 columns will be the attributes derived from the second sample, and so on. So let's take a look at a couple of these. In the very first row, we have a C to G substitution, and the multi-sample report indicates that this particular variant allele has actually been called only in one sample out of the 96 samples that were analyzed in this particular experiment. It then gives details about what is happening at this particular location in each of the 96 samples. So we can see that in the first sample, there are no supporting reads at all for this particular variant allele, even though the coverage in that first sample is 120 at that location. In other words, the first sample does not contain this variant allele. But one of the 96 samples in this list must contain that variant allele. Now if we move on to the second row, we can see that it is a C to T substitution, and that substitution has been found in 50 of the 96 samples. In fact, in the very first sample itself, 41% of the reads which are overlapping this location, so 41% out of the 60 reads which are overlapping this location, support the T variant allele. So if you have 96 samples, then this spreadsheet is going to be extremely large because we have 6 columns for each sample. So we'll have 96 times 6 plus all of these additional columns in this multi-sample variant report. So prioritizing variant alleles when confronted with such a large multi-sample report can be quite challenging. So to see how to go about prioritizing these variant alleles, we'll step back and define the commonly expected criteria. For instance, when would we consider a variant allele to be present in a sample? We would consider it to be present if the coverage is high at that location, if the strand bias is low, and the supporting read percentage is above a particular threshold. Now, the threshold that we would use depends on the experimental criteria. For instance, if you're analyzing cancer samples, 10% might be a reasonable threshold. If you're analyzing normal samples, then the threshold could be 45%, because in the case of a heterozygous allele, we would have approximately 50%, and in the case of a homozygous allele, we would have approximately 95% supporting reads. And if you were doing loss of heterozygosity analysis, we might consider the threshold as high as 90%. So the actual supporting read percentage that we use as a threshold can vary by experimental setup. However, the coverage and strand bias criteria are fairly fixed. Any location which has coverage more than 40 or so can be considered reliable for SNP detection purposes, and as long as the strand bias is within control, say it is less than 20, it can be considered as a fairly decent location. So what we are trying to emphasize in this slide is that coverage and strand bias can be cut off that a user sets once, but the supporting read threshold is something that they would like to play around with and keep varying the supporting read threshold criteria. So with that in mind, suppose we have an allele, a variant allele, and we have the percentage of reads which support that allele in 5 different samples. So let us say there is a specific allele, variant allele, and there are it has 12% supporting reads in sample 1, sample 2 has 61% supporting reads at that location, sample 3 has 48, sample 4 has 43, and sample 5 has 89. So in how many samples would we say that the allele is present? That really depends on what our supporting read threshold is. For instance, if the supporting read threshold is only 10, then we would consider that allele to be present in sample S1, S2, S3, S4, and S5. So overall, we would consider that variant allele to be present in all 5 samples. However, if we were to raise the supporting read threshold to 20, then we would no longer consider that variant allele to be present in sample S1, but we would consider it to be present in samples S2 through S5. So we would say that it is present in 4 samples. So as we keep changing the supporting read threshold, this is what the spreadsheet would look like. When we have a very low threshold, such as 10%, then we would say that it's present in all 5 samples. But as we keep increasing that supporting read threshold, then the number of samples in which we would consider that allele to be present would keep decreasing. And in fact, when you raise the threshold to 90, you can see that that variant allele is absent from all 5 samples. So essentially, we have a relationship between the supporting read threshold and the number of samples in which we would consider the allele to be present. And we could depict that relationship pictorially. So here the x-axis represents the supporting read threshold, and the y-axis indicates whether that allele is present or not. So we can see that the allele can be considered to be present when the supporting read threshold is 10, but it's present in all 5 samples. When the threshold is raised to 20, it is present in only 4 out of the 5 samples, and that is indicated by a slightly less intense shade of green. And when we raise that threshold to 50%, it is present in only 2 out of the 5 samples, and when we raise the threshold to 90, the variant allele is not present in any of the samples. Similarly, suppose we have a second allele which has the supporting read percentage as described out here. It has very low supporting reads in samples S1 and S2, but it has reasonably high number of supporting reads in samples S3 to S5. So what would the picture look like? It would look like this. If the supporting read threshold is 10, the variant allele would be present in only 3 out of the 5. So the color shown out here corresponds to 3. And if you keep increasing the supporting read threshold, the color doesn't change until you reach 50. When you raise the threshold to 50, now we use a lesser shade of green to indicate this variant allele is present in only 2 out of the 5 samples, and so on. So as you're saying, this color indicates that the allele is present in 3 samples at the 30% threshold, and that it's present in only 2 samples at the 60% threshold. So what we have done is to draw pictures which indicate the relationship between supporting read threshold and the number of samples in which the allele is present. We have done that independently for 2 different variant alleles, but what we will have in reality is a large multi-sample report with hundreds of thousands of variant alleles. That is, it will have a lot of rows. So then what would the picture look like? The picture would pretty much look as if you took each one of those individual pictures and put them one on top of the other. So in the next slide, I'll show how to interpret this particular picture. What we have done so far is to show how we would compute the picture. So the picture is reproduced out here, and what it shows is that when the threshold is raised to 30, the height of the bar out here indicates how many alleles can be considered to be significant. So it says that when the supporting read threshold is raised to 30, overall 2 alleles can be considered to be significant, but of the 2, one of them is significant in 4 samples and the other is significant in 3 samples. Similarly, when you raise the supporting read threshold to 50, the height of the bar remains the same, which indicates that 2 alleles continue to remain significant. However, both of them are significant in 2 samples, and that is indicated by the lighter shade of green. So similarly, if we would interpret the last bar to indicate that if you were to raise the supporting read threshold to 90, only one of the alleles can be considered to be significant, and in fact, that is significant in 2 samples. So let's take a look at a real-world example now. Here is an analysis of 96 different HapMap samples. Overall, 3,416 alleles were found to be present in the multi-sample report because this was a targeted resequencing effort where only 384 different amplicons were targeted. So across the 96 samples and the 384 target regions, 3,416 alleles were found, distributed across the samples. Then the question we would have is, what are the alleles which are present in a large fraction of the samples at very high confidence? If you were to choose that criteria, this is what the histogram plot would look like. It shows that if your supporting read threshold is 35, then you would have 204 alleles which you would consider to be significant. Of those 204 alleles, the top portion, which is of the deep intense green, shows that 90 variant alleles are present in at least 80 samples each. Then you have around 40 variant alleles which are present only in 40 to 60 samples, and the remaining are present in 60 to 80 samples. Similarly, the picture indicates that if you were to raise your supporting read percentage to 50, then you'd have approximately 95 variant alleles which you would consider to be significant. And if you were to raise the supporting read percentage all the way to 99, then you'd have around 22 variant alleles which are present in at least 48 out of the 96 samples. So this find significant SNPs workflow and dialog helps you to quickly identify alleles which are common to a lot of samples at high confidence. In fact, multiple filtering criteria are present in the drop-down. The most commonly used one is the first one, which is high confidence common variant alleles. Each one of these filters simply changes the supporting read slider and the number of samples slider. You'll notice out here that we have 2 different filtering dimensions. You could consider an allele to be important if it has a large number of supporting reads and if it's present in a reasonable number of samples. If you would want to adjust both of them. And the different filtering use cases present in the drop-down simply adjust both of these sliders. The first filter, where we choose high confidence common variant alleles, it will show you the alleles which have at least 35% supporting reads in at least 50% of the samples. The second predefined filter is for high confidence rare variant alleles. It will show those variant alleles which have at least 35% supporting reads, but these alleles should be present only in less than 25% of the samples. And finally, if you're interested in cancer biomarkers, percentage of supporting reads may be low, but to have confidence in it, we would require it to be present in a large fraction of the samples, and that is what it translates to. These are just what the predefined filter settings are, but you can freely play around with the slider to change the criteria. So what we have seen so far is how to find significant SNPs when you have a large collection of samples, but the samples are all of the same type. This is what we call experimental setup 1, where you have a collection of samples and you're trying to find SNPs which are present in a large fraction of the samples. We also support other experimental setups. If you're doing matched tumor normal comparisons, then you would use experimental setup 2. If you have different subgroups, let us say you're doing population studies, you have a group of Central East European samples, you have a group of European samples, etc., and you're trying to do group level analysis, then you choose experimental setup 3. And experimental setup 4 is a variation of experimental setup 3, where you're looking for patterns which are present in the group, but which are absent in a predefined normal group. So let me quickly take you through experimental setups 2 and 3. In the case of experimental setup 2, you would have groups. Each group could refer to one patient who has a matched tumor and normal. Within that group, you would have tumor and normal samples. So here we have two groups, one breast and one lung, and within each we have matched tumor normals. What we are interested here is in finding variant alleles which happen to occur only in the tumor sample and not in the normal sample. So one way of doing that is to take a variant allele, see what is the percentage of supporting reads for that variant allele in the tumor, what is the supporting read percentage for that variant allele in the normal, and if the tumor has a lot more supporting reads than the normal, then one would consider that variant allele to be specific to the tumor. So in other words, we need to take a difference of the supporting read percentage between the tumor and the normal. And if that difference is high, then one would consider that variant allele to be present in that group. To do all of that, we choose experimental setup 2, we set up the right interpretation, and then it will compute for each group the difference in the supporting read percentage between the tumor and the normal, and that difference is what is plotted in the view that we have seen earlier. So here we can see that even though there are 604 alleles that are present across the 4 samples, there happens to be only one variant allele which has a significantly higher amount of supporting reads in the tumor than in the normal, and that variant allele is in fact present in only one of the 2 groups. By default, the slider is set at 35. That is because if variant allele happens to be heterozygous in the normal and it's homozygous in the tumor, then the supporting read percentage should be ideally close to 50, but we set it as 35. Similarly, maybe the allele is absent in the normal and it's heterozygous in the tumor, in which case again the difference would be approximately 50, but we set it to 35 to be on the more conservative side. In addition, we have support for population studies where you have multiple subgroups. Here is an in-house experiment where we analyzed 19 different HapMap samples coming from 5 different populations. Then we would set up the interpretation in this manner where we put in the population details in some parameter value out here. Then when we choose experimental setup 3, it allows you to specify the criteria for each individual group in the population. So for instance, out here, we can specify that we are interested in variant alleles which are occurring in at least 40% of the supporting reads, and if they are occurring in all 4 Japanese samples, we are also interested in those variant alleles which are present in all 4 Tuscan samples, the variant alleles present in all 4 European samples, etc. So in other words, we are expressing an interest in only those variant alleles which are present in all members of the population. And we can see the results out here. We can see that there are around approximately 4,000 variant alleles, such that each one of those variant alleles is present in all members of the CEU population, we have some other 5,700 variant alleles which are present in all of the Chinese samples and so on. But there could be overlaps between these groups. For instance, it is possible that the variant alleles that are present in all of the CU samples also happen to be present in the Tuscan sample. To view the relationships between these different allele sets, one can do clustering on the results, and the heat map and the dendrogram out here show the clustering results. Each row in this heat map is one of the 19 samples. Each column is one variant allele, and the color indicates the percentage of supporting reads for that allele in that particular sample. So the red color indicates that the variant allele is homozygous in the sample. The blue color indicates that variant allele is not present in the sample, and the yellow color indicates that the variant allele is heterozygous in that particular sample. So we cluster on the percentage of supporting reads. One can see that the HapMap samples cluster nicely into their predefined populations. You can see that all of the European samples, which are in green, cluster next to each other. We have the CEU samples, Central East European, and we have the Tuscan samples, and those two subtrees are next to each other, which is what one would expect. Similarly, we have the Japanese and the Chinese samples also clustered next to each other. So it forms a nice looking tree which is consistent with our expectations. So in other words, we can use the find significant SNPs to identify high quality SNPs which are consistent in populations, etc. But to view the relationships and the way they're distributed across the different groups, one can use the clustering tool. The clustering tool will also help you to choose any subtree out here and save that list of variant alleles. For instance, if you wanted to look at the variant alleles which are present in a homozygous nature in all of the samples, that would be the subtree out here. One would simply have to click on the subtree and save those variant alleles which are present in a homozygous manner in all of the samples. Similarly, if you are interested in those alleles which are absent in most of the populations, but they seem to be present in the European population, one could save this subtree. That concludes the technical details of our webinar. Thank you.